right, thank you. All right, so to get right into it, my graduate research, my master's and of course now my PhD, has been part of a larger research group within the Mineral Deposit Research Unit at UBC called the Exploration Geochemistry Initiative. This ran from 2015 until very recently. The initiative was started to meet industry demand for better geochemical knowledge and training for young geologists. And $2.5 million was spent over about 5 years to achieve this, run by the late Dr. Peter Winterburn. And the focus was on understanding the superficial processes that contribute to the generation of geochemical anomalies from ore mineralization, and to develop novel exploration strategies and methods in complex exotic surface environments. Of course, one of the most significant challenges I would say facing our industry is the increasing likelihood of new mineral deposits being buried under significant or transported cover sequences. In this figure, we show areas in Canada under cover in the beige with past producing mineral deposits denoted by the red squares. And whether the cover is glacial till, gravel, or soil, we're becoming more reliant on geophysical and geochemical tools and technologies to delineate the mineralization. Of course, this is not just true for Canada. You can certainly extend this issue to other parts of the world. For example, gravel cover in the Atacama Desert in Chile. Unfortunately, existing geochemical techniques often have poor reproducibility and are not always appropriate in certain environments, such as bogs and areas with deep water tables. So the goal of my MSc research was to see if we can try to solve this issue by testing and developing techniques used in environmental microbiology and microbial ecology in the context of mineral exploration in regions of concealed mineralization. So microorganisms influence the environment in a number of different ways, everywhere from catalyzing reactions and in particular the weathering of sulfides at depth to facilitating mineral precipitation and dissolution in the surface soils through their metabolic activity. Microorganisms can also enhance or inhibit geogenic gas formation and contribute to vertical metal migration. There's also a very important relationship between microorganisms and surface vegetation and plants. This relationship can be symbiotic, specifically where we have fungal species that are present at the base of plant roots, and they can protect a plant from harmful metals being absorbed, or they can actually enhance the uptake of important nutrients for life. So the idea of using biology in mineral exploration, it's not that new. Commonly, trees and vegetation can be used as sample media instead of soils to look for anomalous metal content in the right environment. However, using microorganisms is obviously less common, and while the potential, of course, has always been there, for the most part, this type of research has been restricted by the available technology. So some of the earliest studies looking at microbes in the context of economic minerals was focused around spore counting, specifically certain species of bacillus that were thought to be associated with gold. This is literally just counting cells and seeing how abundant they are in like a gram of soil, for example. So some of the more recent work using older methods of DNA analysis looked at genus level taxonomy, looking for abundance differences above different VMS deposits. So the figure on the right here specifically shows abundance of bacteria that perform a specific function. In this case, it's methanotrophy, where the microbes consume methane as a food source, as a carbon source. Mainly the results of these various studies demonstrate that there is potential for using the spatial distribution of microorganisms in soils to resolve anomalies from areas of ore mineralization. There are, however, inherent limitations to these earlier studies of soil microbial community composition, as the techniques that were employed typically show only the most abundant organisms within a community. And really, for a robust application of microbiological based mineral exploration, it's likely that less abundant members of the community are going to be the ones that are sensitive to nearby ore mineralization. So luckily for us, the cost to sequence a single genome has dropped dramatically over the last 20 years, making it much cheaper to analyze sequence data Of course, this is mainly due to the increase in computing power over time, with sequencing technologies exceeding the relationship described by Moore's law, which is, of course, the doubling of computer power every 2 years. With this progression of DNA sequencing technology, our ability to interrogate microbial communities has really just a much higher resolution, where we can now take a detailed look at these communities at the species level of taxonomy. And as this process becomes cheaper, more efficient, there's greater potential for this to actually be applied in exploration programs as it becomes hopefully transformed into a more like field portable based technology. Surface soils are dense with microbial biomass compared to water or deep ocean sediments, for example, making them an ideal sample medium for looking at microbial communities. They're really just, microbes are better geochemists than we are. They'll respond to really subtle changes in their environment that just might go undetected by some geochemical methods that just have innate constraints on their analytical detection limits. So these changes in environment can be abiotic, like pH, EH, moisture, and also biotic, such as surface variation in vegetation that I mentioned, and also predator-prey relationships within soil due to nutrient availability. So a really important concept to understand here when we talk about microbial community responses or shifts or fingerprints is that what we're talking about is the community as a whole. So the metabolic reactions that are taking place are not just carried out by one single organism, but rather it's more of a conveyor belt style of processes with different niches carrying out different functions within the soil. So before I present any data, I just want to take you through the general workflow from sampling through to final data products. So first we sampled B horizon soils with subsequent bacterial DNA extraction from each sample. And next you amplify a marker gene from microorganisms. And this is essentially just creating many copies over and over of these organisms in order for it to be detected by the DNA sequencer. And a marker gene is just a gene that contains variable and conserved regions. So the conserved regions allow for us to identify bacteria using universal detection methods. And the variable regions allow us to assign taxonomy to those specific bacteria. So we can then use this sequencing information to generate species level abundance tables for each sample, as well as assign taxonomy. And then further, we can then look for indicator species within these sort of species tables that can be further assessed for potential spatial anomalies over top of mineralization in the field. So for my master's, I looked at 4 separate field sites, all in glaciated environments. The first 2 being kimberlites within the Slave Craton in the Northwest Territories in Canada. The first is DO18, the more northern location here, located within the Lac de Gras kimberlite field. And the second was the Kelvin kimberlite in the south here. So the first site that I'm going to talk about is DO18. And so, of course, a really important step before interpreting any field data is to have a good superficial geology map and an understanding of how your terrain might impact your results. The field area at DO18 encompasses a vertical kimberlite pipe, so a carrot-shaped intrusion with the surface projection shown in purple here on the left and then in black here on the right. We have a slope break across the middle of the kimberlite with the direction of ice flow going generally to the west. The tilt in the area is variable, but remains consistent sort of above this slope break here, and it can be up to 20 meters in thickness. 4 main pathfinder elements are best spatially associated with the surface projection of DO18, including niobium, nickel, magnesium, and chromium, using 4-acid digests on B horizon soils. And so here we have a map of the normalized sum of those elements with increasing concentration shown in the larger, warmer colored bubbles. So we can see the anomaly here is located on the western portion of the kimberlite with a really nice clastic dispersal train in the direction of glacial movement. So when it comes to the microbiological data, there are many different data products that can be produced from sequence information But we've focused on two specific components that we've adapted for use in mineral exploration. And the first piece of information is related to the composition of the microbial community and how the abundance of different organisms within a community change spatially. We are able to investigate which species are driving these community changes by separating data into different groups in order to find statistical differences. The other type of data that we can extract from microbiological community information is soil biodiversity, which I'll get to a little bit later. So I'm going to first start talking about the microbial community shifts. So for DO18, we separated the data into background and kimberlite. So background, of course, are these blue circles, and then any sample taken above the kimberlite is indicated by these pink stars. And using these particular groups, we queried the data in order to find bacterial species that showed responses based on abundance, specifically related spatially to this kimberlite. And from here on out, I'll refer to those as indicator species. So we separated these indicator species further into positive and negative indicators. The positive indicator species being those species that show an enrichment on top of a buried kimberlite relative to the background materials. So here we have two examples of the positive indicator species found for this kimberlite, with the increasing relative abundance shown with these larger, warmer colored bubbles. And as you can see here, both of these indicator species are showing abundance anomalies pretty much directly above the DO18 kimberlite. You can also plot up the negative indicator species, which will show a depletion above a buried kimberlite or any type of mineralization. In this case, the larger, warmer colored circles indicate a smaller relative abundance, so that the area of the kimberlite is showing a lower relative abundance than the background material. And as with the positive indicators, although not quite as strong as anomaly, we do see it dominant sort of directly over top or on the peripheral margins of the kimberlite. So to put all the indicator species data together, we can plot a normalized sum of both of the positive and the negative indicator species, just like you might do for geochemical indicator or pathfinder elements. As you can see, there's again a distinct microbial community response here directly on top of the kimberlite. And also below the slope break here, even when we have some variability in the superficial material that was sampled. The other type of data that we can extract from community information is the overall microbial diversity of the soil. So competition within communities where there might be, a toxicity effect, or just that certain microorganisms are better adapted to use certain chemical components over others, can contribute to changes in observed biodiversity. So at DO18, we have plotted the number of observed species by the number of reads. And really, as the curve plateaus, you can just be sure that you've sequenced enough genetic material so that you've captured the entire community, or at least most of it. By coloring the background materials in blue here and the materials above the kimberlite in red, you can see graphically already that there's a reduction by about 50% in the number of species above DO18 compared to the surrounding background soils. So we can map that same data set, the microbial diversity, as a map with the larger, warmer colored circles representing that decrease in observed species. And we do see that depression localized dominantly over top of the surface projection of the kimberlite. And so all these data sets that I just discussed, the abundance information and also the diversity, contribute to really a database of information that we can use to identify kimberlites with microbial information. These data products can be added together in an exploration program, including the deposit fingerprint. So that's that unique subset of the microbial community whose change in abundance relative to background occurs directly above kimberlites. The second data set is that background fingerprint. These are those species that are that show a depression in response to buried kimberlites, but are ubiquitous in the surrounding soils. And the third data set is the diversity of the soils overlying the kimberlite. So chemical changes take place in soils due to the presence of a buried kimberlite, changing the conditions enough that different species thrive, but also less members of the microbial community are happy in their environment. And then of course, all this information can be layered together to develop, again, that database on a specific deposit type to then be used elsewhere for other kimberlites. So our work with DO18 showed that microbial community fingerprinting has the potential to delineate concealed kimberlites in up to 20 meters of overburden. And we also found that the communities responded to the kimberlite despite changes in superficial materials, which we saw below that slope right there. So the Kelvin kimberlite, the second one we sampled, has a more regular shape than we had at DO18. A lot of the kimberlites and the kimberlite pipes in the region are shaped like this, and it's thought that just due to pre-existing structures and weakness in the surrounding rocks, the pipe subcrops beneath the lake and plunges down into bedrock as you go northward. The northernmost section is around 150 meters from the surface, and the till in the area is around 4 meters in thickness on average. So here we have a much thinner till blanket, but the added challenge of a thick bedrock host. So with Kelvin, we took a subset of the samples to perform a microbial community analysis. And here we decided to only use information gathered from DO18 to inform our exploration for Kelvin. So really essentially a blind study, which is really more realistic. You're not going to go out and do statistical analysis on something where you don't know where the kimberlite is. So first we looked at microbial diversity, where we did see a response in the soils above Kelvin, with the larger circles representing that lower microbial diversity, so that depression. And we do see that depression localized over top of the northern part of the pipe, showing that microbial diversity shifts are indeed linked to some component of the buried kimberlite here. And then after looking at diversity, we decided to look for indicators that we had at DO18 in the Kelvin data to see if they were conserved. We had anomalies conserved across field sites. And we do see that. We see many of the DO18 indicators present here, showing an increased relative abundance directly over top of the kimberlite. And so these are just two examples of those positive indicator species that show anomalies in that same area, of course, as the low diversity in the northern section of the kimberlite, which I'll point out is actually the deepest part from surface. And then here are two examples of the negative indicator species. So recall, these negative indicators are those that have a depletion on top of a buried kimberlite. Again, these are localized to the northernmost section and do show a weaker anomalous signature, which is the same type of stuff that we saw at DO18. So just like at DO18, we can map the normalized sum of all of the positive and the negative indicator species from this site to produce a very, very clear anomaly with very little background noise, at least in the samples that we've analyzed. So not only did we have direct kimberlite detection through 20 meters of till with DO18, but the results were reproducible at Kelvin with some of the same indicator species present at both sites. And in addition, direct identification at Kelvin was successful through up to 150 meters of bedrock. Because we've also conducted geochemical studies at both DO18 and Kelvin, we were able to compare our microbial exploration technique with that of traditional soil geochemistry. So on the top row, we have the normalized sum maps for the microbial indicator species. And in the bottom row, we have the sum maps for nickel, niobium, magnesium, and chromium. And so comparing these maps, I think you can appreciate the accuracy, at least with these data sets, in identifying these kimberlites with microbial community data versus geochemical methods, especially at the Kelvin kimberlite. There are also a greater number of potential indicator species than indicator or pathfinder elements, where you're limited by the finite number of elements on the periodic table. Microbial communities are made up of thousands upon thousands of extremely sensitive taxa that can be used potentially to find concealed kimberlites, or really any type of mineralization for that matter. So the next two field sites are porphyry copper deposits. They are located in the intermontane belt of the Canadian Cordillera in British Columbia. So the Deerhorn porphyry is characterized dominantly by copper and gold mineralization. And I'm sure many of you are familiar with the Highland Valley Tech property. And so we sampled the Highmont South deposit, which is dominantly made up of copper and molybdenum mineralization. So at Highland Valley, the first one we looked at, the soils that were sampled came from a relatively uniform till blanket with an average thickness of around 5 meters over top of the mineralization. The most recent direction of glacial movement in this region has been determined to be kind of subparallel to this this more elongate body in the middle. There's also a system of faults present, with one cross-cutting the mineralized body that have a shallow northeast-southwest trend. Mineralization in this case at the surface projection is characterized by 0.1% copper equivalent in the black line. So 4 elements are best spatially associated with the mineralization. So we have copper and molybdenum on the top row and silver and bismuth on the bottom row. These anomalies are mostly localized to this larger elongate body in the middle, but you can see some anomalies spatially correlated with the faults as well. We got an anomaly here, another one over here with copper. As with the kimberlites, so for the microbial data, I performed an indicator species analysis at High Mountain South and found several positive indicator species and anomalies associated with the porphyry mineralization. So here we have 2 examples of those, and so those are on top, with the sum of all the positive indicator indicator species showing an anomaly, mostly in this elongate body, but we also have one in the smaller section, and then we also see this highlighting along the faults as well. We also found those species that are depleted in relative abundance over top of the deposit, which still show an anomalous values above mineralization, but it's not quite as nice an anomaly compared to the positive indicator species. I'll talk a little bit about why we think that this might be the case in a couple of slides. Not that there aren't anomalies on top of mineralization, but we have a lot more noise going on in the background data. So the Deerhorn copper gold porphyry is overlain by around 170 meters thick till cover in the northwest to less than 10 meters in the southeast. The area is characterized by lots of swamps and organic deposits. And in this case, the mineralization is characterized by 0.2% gold equivalent. And that's this sort of black wiggly line defining mineralization there. Ice direction, most recent ice direction is going this way, but we do have historically glacial movement, evidence for glacial movement, also going anti-parallel to this. So each element in this case was normalized to organic carbon just because of the swampy and organic nature of the material sampled. We see copper and molybdenum here. We do see anomalies at the Deerhorn mineralization at this area, but these geochemical anomalies have not developed with great spatial correlation to mineralization. We're mostly seeing anomalous samples kind of in the background material here, or sort of at the periphery of mineralization. And again, nothing super strong at Deerhorn. We can also see potassium, arsenic, and they also, I would say, are going along the same trend as the other 2. We really don't see really strong geochemical anomalies here, and in particular with potassium even sort of in the background. So with the indicator species analysis, the positive indicator species resolved the deposit actually quite well here, especially given that we didn't see a very significant geochemical anomaly crop up here. And again, we have those increased relative abundances occurring directly over top of mineralization. And considering the materials sampled, the noise is actually pretty low. Again, same with Highland Valley, we have these negative indicator species, and we do see a correlation, but it's certainly more erratic. Again, just like we saw at Highland Valley, but certainly I would say a stronger anomaly than some of the geochemical data. So in both cases at the porphyry sites, we're thinking that some of this noise in the negatives is basically present because we're not accurately sampling background material. So porphyry copper mineralization is gradational compared to kimberlites. You see that really strong lithological control, and the porphyry footprints are quite a lot bigger. So we are thinking that if perhaps you were able to actually extend out into background a lot further, or even take like a set of background samples for reference, you might see some of that noise get reduced in the negative indicator species. But we also, because of the nature of sulfide mineralization, because of that gradational nature, these equivalents, these outlines of mineralization are not necessarily reflective of the specific mineralization. Obviously, you can have a very gradational zone, and so it would be interesting to see like how the microbial anomalies potentially correlate with a gradation outwards, depending on the type of sulfide mineralization that you have. So at both kimberlite sites and porphyry copper deposits, I think I've shown that there is a strong potential for microbiological based mineral exploration methods, hopefully to be continually developed through cover mineral exploration. Differences in microbial community composition are readily discernible through indicator species analyses at both kimberlite and porphyry copper deposits. And we've also seen, like I said, differences in the application of the kimberlites compared to the porphyries, namely, like I said, in the quality of the negative indicators at both Highland Valley and at Deerhorn. And I think that's really important to recognize the limitations when applying any technique for the purpose of mineral exploration. Like I said, with that gradational nature of mineralization, that's something where you would always want to take that into account, or potentially even on our end, developing these types of techniques, thinking about how we might best apply these things, in sulfide environments. Are we just going to use the positive, negative, or positive indicators? Are the negatives going to give you more noise? And we can certainly leave those out. And that's just all part of developing a new technique like this, I think. So the majority of my MSc work has really been surrounding field test cases. These types of studies are super valuable, obviously, as we've seen, and there will be a focus on continuing to have deposit scale investigations, specifically related to how we can use existing databases to predict chemistry at specific mineralization types. However, these studies thus far are descriptive in nature, and we do want to expand existing microbiological toolbox to ask deeper questions about soil, about microbes, and how they relate to economic geology. So the first one, and I would say one of the more important ones, I'll be looking at the functional potential of microbial communities and how microbial communities respond to changes in temperature in northern environments in the soil. And so this is really important on the mineral exploration side, because we want to know how much climate change in the north is actually going to impact our microbial anomalies, and to speak to the longevity of our indicator microorganism databases. So in 10 to 15 years time, are those still going to be useful? By looking at the functional potential, we can also look at carbon cycling in northern environments and assess the feedback between microbes and greenhouse gases. So this type of information is valuable when informing climate models to help make predictions on whether different types of soils will act as carbon sinks or sources with increasing temperatures. So I will also be assessing the same types of things in bogs. So peatland bog systems also remain a significant challenge, I think, when exploring for mineral deposits, as very few techniques are marketed for application in those environments. High degrees of saturation and organics change the mobility of elements and potentially skewing a geochemical signal from mineralization. Bogs are also crucial to carbon sinks, globally, and I'll be looking at the role of microorganisms in those environments. The last two objectives are experimental in nature. The first I'll be looking at is how microorganisms actually sense and respond and use minerals and water. So for this, I'll be using a model E. coli organism and exposing it to different minerals, some sulfides, but some rock-forming minerals as well, and looking at their gene regulation and what metabolic processes are actually activated. So what kinds of things are they doing in order to get at some of the nutrients potentially in these minerals. So the second is a more practical, I would say, experiment. So currently our standard procedure has been to freeze samples as soon as possible. Obviously, depending on the location of your exploration program and access to transport, samples could be sitting out for a pretty long time. Now with our current work, like especially the Kelvin work, some of our samples sat out for weeks outside during transport back to Vancouver. Based on the data that we have, as you saw, we still get pretty striking anomalies, but we do want to test this empirically because until a sample is frozen, there's always potential for continued microbial activity or cell lysine, and that may impact the composition and relative abundance of some of our indicator organisms. So this experiment is pretty simple. It involves leaving soils in containers over a long period of time, about a year, and progressively sampling it to look for changes in microbial community composition. So some of our treatments right now are keeping the soil like in a bag, just at room temperature, keeping a bag open to room temperature, heating it to 60 degrees, and then leaving it at room temperature, and then also storing them in the freezer at -20 degrees, and then in the fridge at 4 degrees. So that work is ongoing, and hopefully we'll have some results from that in the next couple of months, and then a more long-term result in about a year. So I just talked a lot about metabolic potential and activity and gene regulation. So what does that actually mean? To understand how we assess activity and assign function, we really just need to look at the central dogma of microbiology. So this describes how instructions from DNA travel at the cellular level to inform how an organism actually behaves. So collectively, the study of DNA and RNA is called genomics, whereby we look at genes themselves. This can involve looking at a marker gene in order to assign a sequence of DNA to a specific species, which is what we have done with the data here that I presented. Or you can do something called metagenomics, whereby you investigate genes from an organism and see what reactions and metabolomes they might have the potential to carry out based on their genetics. And then on the other end, we have study of proteins and metabolites. And so this goes a step further to look at what the actual activity of a community is in an environmental sample. So not just the potential activity based on genetic information. So what are they producing that actually proves that they're carrying out a certain metabolic pathway? And so integrating these types of analyses would greatly expand our knowledge on the relationships between microbial communities and mineral anomalies, and to support the use of these methods for future through cover mineral exploration. So I hope today that I've illustrated that there really is a lot of potential for discovery and innovation when it comes to geomicrobiology, whether your focus lies in mineral exploration or on the environmental side. Obviously, the use of modern DNA sequencing is quite new for the exploration space, and we've done our best to keep it as transparent as possible. But people have been using microorganisms really to remediate acid rock drainage and hydrocarbon contamination for quite a long time now. A few years back, I was actually at a mine in South Africa, and they used microbial communities to recover gold instead of roasting, which was pretty cool. Up in Canada in Yellowknife, we have a lot of issues with arsenic and roasting that historically has gone up there. So it would be really neat if you could use microbes to extract that pretty cleanly. So there really is a lot of opportunity in this space, and collaboration really is the name of the game here. So I would just like to acknowledge the whole team, which consists of geologists, geochemists, microbiologists, and especially the students who have made this project successful. And thank you.